The electron transport chain occurs in the mitochondria, the powerhouse of the cell, composed by an outer membrane and an inner membrane. Inside this, there is a space, inside this purple line called the matrix. The outer mitochondrial membrane is porous and is permeable to small molecules. The inner membrane, on the other hand, is composed by a phospholipid bilayer, which makes it impermeable to small molecules and ions, including protons. This creates a space between the two called the intermembrane space. Within the inner membrane, there's a matrix with few protons. There is a high proton concentration in the intermembrane space, giving it a low pH, as opposed to the matrix, which has a low proton concentration. This difference in concentration of protons is created by the electron transport chain. This gradient provides electrochemical potential, which ultimately provides the energy needed by the ATP synthase for the phosphorylation of ADP, a process which is highly thermodynamically unfavorable.
In the inner mitochondrial membrane, we have the electron transport chain. We have complex 1, which is the NADH dehydrogenase. We have complex 2, called succinate dehydrogenase, complex 3, which is cytochrome C oxidoreductase, and complex 4, cytochrome C oxidase.
There are 5 electron carriers used in this electron transport chain. NADH can carry 2 electrons at a time, flavoproteins can carry 1 to 2 electrons at a time, whereas iron-sulfur proteins can only carry 1 at a time. Finally, we have the mobile electron carriers, ubiquinone, which can carry 2 electrons at a time. It has 3 forms. The fully oxidized form has no electrons, the intermediate has 1, and the fully reduced 1 ubiquinol has 2. It can collect electrons from complex 1 and 2 and bring them to complex 3. Cytochrome C can carry 1 electron from complex 3 to complex 4. And now, magic.
NADH will become oxidized into NAD+, FMN will take its 2 electrons. Then the iron-sulfur center will bring the electrons to Q one at a time. From the matrix, 2 protons will join Q forming QH2 and 4 protons will be pumped out. Complex 2, or succinate dehydrogenase, is an enzyme from the Krebs cycle. Succinate will bind to a binding site and will be transformed into fumarate. 2 electrons will be passed to FAD. The iron-sulfur center will pass 2 electrons to Q one at a time, forming QH2.
This time, no electrons will be pumped out.
QH2, coming from complex 1 or 2, will go to the binding site of complex 3, also called cytochrome C oxidoreductase. It will donate its 2 electrons, 1 each, to cytochrome C. Q goes back to its fully oxidized version. 2 protons will be used in this process, and 4 protons will be pumped out into the intermembrane space. The 2 mobile cytochrome Cs will bind to complex 4 cytochrome C oxidase, and the oxygen will attract the 2 electrons along with 2 protons to reduce oxygen to H2O. From the matrix, 2 protons will be pumped out into the intermembrane space.
So in this video, we have used 2 cytochrome C. In this way, we have only used half an oxygen, which with 2 protons and 2 electrons yields 1 water molecule. Remember that from 1 QH2 you get 2 reduced cytochrome Cs. But in reality, to get a full oxygen molecules, you need 2 QH2s and 4 reduced cytochrome Cs. Therefore, with the use of 4 protons and 4 electrons, you will get 2 water molecules. When electrons enter the electron transfer chain through complex 1, a proton pump using NADH, 4 protons are pumped out. Complex 3 pumps out 4 protons and complex 4 pumps out 2. This gives a final balance of 10 protons, which in the inner membrane space can be used to make 2.5 ATPs. When electrons enter through complex 2, where FADH2 is used, no protons will be pumped out. Complex 3 pumps 4 protons out, and complex 4 pumps 2 out for an overall balance of 6, which can be used to generate 1.5 ATPs.